Hello, everyone, and welcome to today's webinar, where we'll be discussing considerations for chromatographic method development. My name is Jennifer Caveney, and I will be facilitating today's presentation. I would like to introduce our two speakers today. Our first speaker is Dr. Jamie Willems. With a PhD from the University of Saskatchewan, Jamie brings a wealth of expertise in food chemistry. As the manager of special analysis at Eurofins SFA Laboratories in New Berlin, Wisconsin, she leads cutting-edge work in chromatographic techniques across a variety of instrumentation types. Her deep experience spans custom method transfers, method development, suitability studies, validation protocols, and the nuanced world of flavor and fragrance analysis. Our second speaker today is Lily Zafis. Lily holds a master's degree in food science from the University of Saskatchewan and serves as supervisor of special analysis at Eurofins SF Analytical Laboratories in New Berlin, Wisconsin. With a sharp focus on liquid and gas chromatography, Lily tackles complex analytical challenges from contamination investigations and custom analyte method development to off-odor, off-flavor analysis and form material identification. Her work bridges precision science with real-world problem solving. Jamie, whenever you're ready to begin.
Sounds good, thank you. So thank you for that introduction. As mentioned, I'm Dr. Jamie Willems, and today alongside with my colleague, Lily Zafis, we will be presenting on considerations for chromatographic method development in food, beverages, ingredients, excipients, and dietary supplements. So today I'm just going to start with a short introduction to method development and method validation, discuss considerations when developing a new method, and then I will pass the presentation over to Lily to discuss considerations for instrument choice, as well as going over a few case studies on method development. So why do we care about method development and validation? Well, I imagine if you are here listening to this presentation, you're already aware of the importance of method development and validation. However, method development is required for any sample type, such as a new matrix or analyte that cannot be analyzed using an existing validated method. Method development can range from small changes to a method, such as adapting a sample preparation of an existing method, to starting completely from scratch with an entirely new approach. For example, you may have an existing method for the analysis of an analyte or compound of interest in a beverage, say, for example, sucrose. However, you have developed a new product, such as a chocolate and peanut butter-based material, that also needs sucrose analysis. In this case, you may be able to adapt your current method with new sample cleanup and preparation steps, or you may find that now you have interferences and need to find a different approach for this matrix. Another example would be you have a new analyte or material for which there is no compendial or commercial offering for this compound. This could be due to the compound being a novel compound, for example, a new pharmaceutical, or it could be due to changes in testing requirements, such as regulatory changes that require lowering detection limits. In this case, you might not have a current method you can adapt, but would need to start fresh. It is not unusual for method development to involve work on multiple approaches simultaneously before eventually choosing the best fit. Following method development, method validation is typically performed. Method validation is used to demonstrate that an analytical procedure is suitable for its intended use. Reliability or lack of reliable data produced from an analytical method can have widespread impacts on health and safety. The most obvious example of this is in the pharmaceutical industry, where an unreliable method may lead to incorrect dosing of an active ingredient or potentially harmful impurities being present. But this can also impact other areas, for example, the food industry, leading to misleading label claims or issues with product consistency. For example, if you have a product that needs a specific color and you need to measure the content of that color in your product, if your method is unreliable, you may have inconsistent product quality, with some batches being too bright or others being too muted in color. So before we get into a deep dive on method development, I want to give a very brief introduction into method validations and verifications. Typically, this step follows method development as a way of proving a method is fit for the purpose and giving an indication on how it will perform on a routine basis in your laboratory. So method validation or verification is the process of establishing the required performance criteria of a new, updated, or newly implemented method. In this way, we can show that a method is performing as intended, or fit for purpose. Fit for purpose is a phrase you may hear often when considering method development and validation. When we talk about fit for purpose, basically we are talking about a method meeting the needs of those who use it. For example, you may need to run an assay where the exact concentration of an analyte is very critical. The difference of a couple part per million may be significant. In this case, you want to set your method development goals and your validation criteria to reflect that need. For example, you may need an accuracy with percent recoveries ranging from, say, 98 to 102%. Or in some instances, you may have an impurity for which you just need to know general amounts. The difference between 10 to 12 ppm isn't significant, but the difference between, say, 10 and 100 ppm is. In this case, super tight acceptance criteria, as the example before, is unnecessary, and they can lead to needless extra work, both in method development as well as in routine application. And this extra work obviously can lead to additional time and monetary investment at all stages of development and use of the method. Another important distinction when we're talking about method development and validation is the difference between method validation and method verification. I won't go into too much detail here as this is not really the main focus of this presentation, but generally speaking, method validation is a more robust process involving a greater number of criteria and is typically carried out across multiple days and possibly even multiple instruments or laboratories as needed. Verification tends to be a simpler process used to demonstrate that an already validated method performs as intended in a new environment or after a more minor change. Method validation is typically used when you have developed a completely new method or made significant changes to an existing method. For example, changing equipment, such as from a flame ionization detector to a mass spectrometer, or adding in a new sample matrix type. Verifications are performed when you are implementing a method that is already validated, perhaps a method transfer from another lab, or implementation of a compendial method with performance data, or making simpler, smaller changes to a method. There's not a strict line between method validation and verification, and each project should be evaluated on a case-by-case basis. So why do we validate or verify our methods? So besides our own desire to have a working method, it is due to regulations. Many regulatory bodies require method validation for use in release and related testing in order to prove the integrity of the data. These groups, such as the USP, the FDA, and AOAC, also typically provide great resources on method validation and verification, and are a great place to look when planning your own developments and validations, especially if you need a method for use with one of these regulatory bodies. For example, if you have FDA release testing, you would want to look at the FDA criteria to make sure your method meets the needs and their specific requirements. So now you know why we validate. But before you can even get to that step, you need the method. If you don't have a method to begin with, where do you even start? I'm going to walk through the basis of how to approach method development if you are new to the process, before handing off to Lily to go into more details. Specifically today, we're going to be focusing on chromatographic methods, but you can apply these basic principles to any type of method development. So where do we start? Well, the first step is to determine what information you exactly need from the method you are developing. Will this be an assay for a major component and you require an exact quantifiable number? Is this a screening method for impurities where a detect, non-detect result is enough? That is it a qualitative method good enough, or do you need a quantitative result? What sort of concentrations do you expect to see, or what detection limit do you need? The answers to these questions will help you define a number of factors about your method that help focus your development and select the appropriate technique. For example, if you're analyzing a major component, say sucrose again, that you expect to be present in high concentrations, for example, in a very sugary beverage, maybe 5 or 10% of the total product, then you don't really need to worry about detection limits. You can choose a less sensitive detector that may be less expensive, easier to run and maintain, more widely available, and so on. However, if you're looking for trace impurities in the parts per million or even parts per billion levels, you'll want to focus on more sensitive and selective approaches. Similarly, if you're looking for a quantitative result, you will want to consider factors such as the linear range of your method, calibration curves, and additional suitability, such as check standards and precision requirements. So once you've solidified what you need from your method, that is what output you are requiring, the next step is to consider the analyte of interest. This will help determine which approaches are even viable. For example, if your compound is a volatile, I would first look at gas chromatography-based methods versus non-volatile components, where you would consider liquid chromatography. Additionally, the structure affects what detectors are viable and their sensitivity. For example, some compounds are very UV active, such as those with lots of conjugated double bonds, while others give little to no response by UV, such as those like alkanes, long-chain fatty acids that don't have these conjugated groups. And therefore, they'll give little to no response by UV, and another approach is required, so you can use a UV detector in this case. This is also a good place to consider sample matrix as well. For example, maybe your matrix contains a lot of other UV active compounds that may interfere with your analyte of interest. In this case, you may consider a more targeted approach, such as using mass spectrometry, or look into sample cleanup to remove the interfering components. It is important to note that a lot of development decisions are based not only on what is ideal, but also what is available. For example, if your lab only has a gas chromatograph and your analyte is non-volatile, you may will need to look into derivatization approaches to make the analyte volatile and work on the instrument you have. On the whole, there is no one-size-fits-all approach to method development. The important factor is that you look for an approach that takes what you need from the method and what is available to you in consideration, that is fit for purpose. So on this slide, I have a number of parameters that are important to consider when developing and running feasibility on a method. These are the same categories you will see covered in method validation and verification. So it is important to consider each category and determine what is needed for the method you are working on. I've briefly touched on each of these in previous slides as examples, so won't go into depth here, but these categories are a good place to start when outlining what you need from a method. As discussed, factors such as detection limit, accuracy, selectivity, and so on. So now that you have determined what kind of method you need, for example, an assay versus impurity, quantitative versus qualitative, what type of instrumentation will work with your analyte of interest, and what suitability parameters are important, and what criteria you need to hit, how do you approach actually developing the method if you have never done this before? Well, the best way I've found to approach these problems is to look and see what others have done. Some may call that cheating, others might call it standing on the shoulders of giants. But building off of already established methods for similar analytes or matrices is the fastest way to hit the ground running when developing a new method. Start by looking for methods that have already been validated. For example, compendial methods from sources such as USP or AOAC, or other methods already being run in your laboratory, and so on. If you can't find a validated method, scientific literature is another great resource. They are typically on the cutting edge of method development. However, many of these methods do not establish performance criteria and are rarely validated. Finally, institutional knowledge is a great resource. Don't be afraid to talk to people more experienced than yourself. Use past methods as a chance to learn and build off of. Once you have established your approach, the next step is method feasibility. And this is testing to see that your desired approach actually works. Feasibility should be used to determine how a method performs and to help set appropriate performance criteria for future validation. You will want to set up your method, show that it works on your analyte of interest. That is, are you getting the desired separation, detection, and so on. You'll want to test factors such as reproducibility. Does running across multiple days, multiple preps give you the same result? You want to look for things like accuracy. If I add in a known amount of my analyte, can I get that back? What kind of percent recoveries do I get through spiking studies? Look at factors such as robustness, detection limits, linearity, and so on. Often during this stage, you'll find that you'll need to go back and reevaluate and redevelop your method. Rarely are you going to get it right on the first try. Often you're going to be reiterating, sometimes repeatedly, until you get a method that performs as desired. And so as you do this, you might also find that you want to reassess your desired criteria to be more realistic while still being fit for purpose. We all want that perfect method, but unfortunately, science is rarely so kind. And finally, once you're happy with your method, you'll want to write, if you need a validated method, you'll want to write a validation protocol using all the information you've gained from feasibility. It is important to write your protocol and establish your criteria prior to actually running the validation. And then the final piece of the puzzle is to validate your method. Perform your analysis as per your protocol and document your results to determine if they meet the specified criteria. If you pass, congrats, you have now validated your method and you're good to run on a routine basis. However, if it fails, don't throw away the data. Analyze it for what went wrong and use that knowledge to return to feasibility and continue to refine your method until you have one that is robust and repeatable.
Okay, now that we've gone over initial considerations for method development, I'm going to talk about chromatographic instrumentation options and how to choose a system that will best suit your needs. Our first system is a gas chromatography system with flame ionization detection, or GC-FID. This type of detector is a good match for carbon-containing analytes, which are combusted in a hydrogen flame and produce ions that are measured. The best features of this type of system is that they are widely available and straightforward to run and maintain. They are also reliable, often sensitive, and typically have a wide linear range. This detector is considered universal in terms of carbon-containing or organic analytes. However, there's no truly universal detector, though it is a common phrase for detectors that can respond to many compounds. The drawbacks to a GC-FID is that it lacks selectivity. So the FID will respond to any carbon-containing molecule in your sample, including your solvents. Additionally, there are times where an FID does not have the sensitivity of a targeted detector, though this will be analyte specific. As an example, this chromatogram on the right is of a relatively pure analyte, so you can see there aren't many other extraneous peaks besides the analyte at about 8.5 minutes. However, as I mentioned, an FID will also respond to many solvents, so the peak here between 2 and 4 minutes is our ethyl acetate solvent peak. Our next system is GC with mass spectrometry. For this system, compounds are separated by GC and then ionized and identified by a mass to charge ratio. This can be run in tandem or single. To expand on that, running an MS in single quadrupole mode would involve scanning a mass range throughout your method. The resulting chromatogram is then a total ion chromatogram, or a TIC, which shows intensity of total ions at any given time. To take mass spec to the next level, you could use tandem mass spec, which monitors a single ion transition, which is the single mass as it breaks into another mass, and this gives a very high level of selectivity. Mass spec detectors are typically very sensitive. They are generally considered universal, though this will also be based on the specific ionization properties of your analyte. The biggest benefits of the mass spec is the advanced level of selectivity that they provide, which can be used for compound identification. As an example, here I have a GC-MS total ion chromatogram for an essential oil sample on the right. This peak that is marked with an arrow has the mass spectrum or ions at the retention time, which match to a library reference shown on the right for 2-bordenone. When running GC-MS, analytes will ionize and fragment in a very specific and repeatable way, meaning that across systems and methods, compounds can be identified with great confidence without any other information besides the ions that they are producing. However, it isn't only good things here, and the things that make this detector great are the same things that can cause problems. You are more likely to see interfering substances in cases like this, and additionally, you also do need to have a more experienced analyst typically to maintain and run this kind of system. For our next system, we're moving into the liquid chromatography world, and starting with the simplest and most common detector generally, which is a UV-Vis or photodiode array. This detector is good for compounds containing chromophores, which absorb light at a specific wavelength, as Jamie described earlier. This detector is good for these detectors are known to be sensitive and targeted, since you can scan at a selected wavelength that is highly responsive to your analyte. In an ideal world, your matrix will not respond at that wavelength. If you are running with photodiode array, this means you can scan at a range of wavelengths throughout your run, which gives you an absorbance spectrum for each peak, which will be specific to your analyte. So in this way, a PDA detector can be highly selective and used for identification. These detectors are also common and inexpensive and typically have a wide linear range. The only major drawback is that your analyte must contain a chromophore, otherwise you simply can't detect it using this type of system. As an example, the chromatogram on the right is showing an overlay of 4 different monitored wavelengths for the compound sunset yellow, otherwise known as yellow 6, which is a common food dye. For this method, we used PDA scanning, so what I'm showing here next to the peak is the unique UV-Vis spectrum for this compound, which you can then use to confirm the identification in a sample. Our next system is LC with refractive index detection, or RI. In this system, the refractive index of a reference cell containing mobile phase is constantly compared to the refractive index of the mobile phase as it passes through the column. When an analyte is present, there will be a change in that refractive index, which is then measured. Perks of this detector is that it is generally universal, as most compounds will cause a change in refractive index, and these systems are inexpensive and common. However, the drawbacks are generally low sensitivity, no selectivity, and you also cannot run gradients on the system because the refractive index of your mobile phase would change throughout the run. These detectors are also sensitive to the environment, such as changes in temperature. RA detection is more common in older methods and isn't as widely used in new developments, often because of these drawbacks. However, it's still the primary choice for specific compounds such as sugar analysis. The chromatogram I'm showing on the right is the sugar profile of an ice cream sample which was extracted with water. You can see fructose, glucose, and sucrose, and additionally, you can see a peak here at 3 minutes in the chromatogram, which is the sample extraction solvent, in this case, which I said was water, since our mobile phase system was an acetonitrile water mixture. Our next system is LC with evaporative light scattering detector, or ELSD. In this system, the mobile phase is nebulized and then evaporated, and the analyte particles are passed through a beam of light, where the amount of light scattered is measured. These detectors are often thought of as alternatives to RI, as they are generally more sensitive and are also compatible with gradients. The drawbacks are that as a universal detector with increased sensitivity, matrix interference can be an issue. In addition, non-volatile buffers cannot be used, and the systems often have a narrow linear range or must use a log-log linear curve. The chromatogram I'm showing here is the most common ELSD method that we run in our lab, which is the assay for taurine from USP. Our final system today is LC with mass spectrometry, either single or tandem. Compared to GC, LC-MS can use a variety of ionization techniques and solvent systems for separation. Because of this, the identification isn't always as straightforward. As an example, the primary ion you might expect for your analyte might end up being M plus H, M plus ammonia, M plus sodium, or it could form an adduct with your mobile phase additive. Because of this, systems with high resolution are often necessary for identification, as these can collect accurate mass data, which can then be matched to known compounds. Drawbacks to LC-MS is that you can't use non-volatile buffers, as with ELSD, and it can have issues with matrix interference. Additionally, as with GC-MS, it can be expensive and complex to maintain and develop methods on these systems. The chromatogram I'm showing here is from a tandem mass spec method we developed for the analysis of capsaicin and dihydrocapsaicin. And what I'm showing for the DHC peak here is the response of the specific ion transitions that we monitored in this case. We're now going to transition into some real-world case studies where we developed and validated methods with a variety of goals and platforms. Our first case study is for the analysis of a novel health molecule in a smoothie beverage formulation. So going back to what Jamie talked about earlier, the first thing we did is consider what our goals for the analysis were. In this case, we were looking at quantitation at the weight % level in a final product, so essentially an assay. We also knew we were looking at a complex smoothie matrix with many potential interference. And we were working with a novel molecule, so building off of existing methods or literature was not going to be possible. Based on this assessment of the molecule structure, which was that it was not UV active but was organic and had good volatility, and considering the goal of keeping future analysis cost effective for our client, GC-FID was selected for our platform. During feasibility, we first optimized our instrument conditions to have good peak shape and the desired detection level, which in this case was not very low since we were looking at assay levels. We also tested a variety of solvents and did spiking studies to see if we could reduce how much matrix was being extracted. We also tested several internal standards as well in this process. And the chromatogram on the right was the final conditions for our method, which shows the internal standard and analyte at 4.6 and 7.8 minutes respectively. The main challenges during this process was the complexity of the matrix we were expecting. In addition to running final product precision samples, we obtained a blank smoothie matrix which did not contain the active ingredient so that we knew our accuracy study would be as effective as possible. The other major challenge was the novelty of the molecule. We couldn't get commercial standards for this, and we did have to rely on a client-provided standard for our analysis. So to account for this, we used the same GC method, but we used mass spec detection during our validation in order to demonstrate the specificity of the molecule using its fragmentation. Our next case study is analysis of a trace impurity in an artificial sweetener. Here we were looking at the opposite of main component of analysis. We were looking for low levels, which would be less than 0.1% in the sweetener product. Based on the structure and expected concentration, we selected liquid chromatography with mass spectrometry for this method. Our feasibility included chromatographic optimization, along with a focus on the mass spec ionization parameters to ensure that we could achieve the detection level that we needed. We also experimented with the extraction to ensure that the main component wouldn't cause interference with analysis of our analyte. The chromatograms here are showing a low-level standard at 0.02 ppm on top, and then the bottom is a 0.5 ppm of our impurity. Our initial challenge during this development was achieving the sensitivity that we needed. Our analyte ionized in negative mode, and if any of you are familiar with mass spec already, you'll know that generally speaking, negative mode is much less sensitive than positive mode. So we did have to optimize our ionization conditions such as cone voltage in order to get ionization at the level that we needed. We also found that the analyte had more fluctuation in day-to-day signal than we had expected. So even though our sample runs were consistent, we were seeing variation in signal that shifted our linear range between runs. So when we did write our SOP for this and our validation criteria, we had to consider this and make sure that we were fitting the purpose of the analysis while still allowing for that day-to-day flexibility in ionization. Our next case study is for 34 trace drug residues in wastewater. So for this method, we were shifting from single component analysis to a screening approach, and we wanted to see truly trace amounts in the realm of PPB detection and quantitation. We also needed to cover a range of drug residues which in a variety of structural classes. Based on the large number of analytes, their variation in structure, and the desired level of detection, the most promising platform was HPLC-MS, as this is the most common method for this type of broad low-level drug screen. Feasibility included identifying and optimizing unique ion transitions for each analyte and ensuring that we had clean baselines to achieve the low detection and high specificity. We also tested a variety of isotopically labeled internal standards for each drug class. The chromatogram on the right is showing all 34 analytes and their internal standards at 50 parts per billion. So you can see that the ionization intensities and peak shapes vary significantly for the different types of drugs. So this is why it was important to have internal standards that would ionize in the same way as their respective drug class. The final part of feasibility was modifying the chromatographic parameters to get ideal peak shape and separation for as many of these analytes as possible. And when you run LC-MS, you don't technically need separation as long as you have unique transitions, but it's always ideal to get as much separation as possible. The challenges here, as I described, were the optimization for a variety of analytes, which ended up being a large investment in time. The other factor was determining validation guidelines. Since our goal was very low-level quantitation and detection, we relied heavily on EPA and FDA guidelines to ensure that we were meeting that purpose while creating criteria that could pass at those low levels. So for example, a big part of our validation was replicate sample spikes at a 10 part per billion level. But our acceptance criteria had a wide range because this is such a low level to be analyzing. For example, if you had a recovery of 13 parts per billion for that spike, that is 130%, which in many other circumstances would not be considered acceptable. But when the true value was 10 parts per billion, you realize that those two numbers are not significantly different from each other. And that would be well within the accuracy that you could expect in that range, and it still demonstrates the performance of the method which fit our purpose. Our final case study here is the identification of 9 FD&C approved dyes. The goal of this analysis was to qualitatively identify these food, drug, and cosmetic approved dyes, which is different than the other case studies, which were all quantitative. And additionally, we wanted to develop this method and leave room for future matrix extensions since there are so many different potential applications for this screen. Based on our analytes being dye molecules with known absorbances, we selected LC-UV as our platform. For feasibility, we optimized our chromatography to have clear separation and strong UV-Vis spectra for each analyte. We also did a variety of test extractions and purifications on different foods to see how the method would perform in different matrices. The challenges for this method was to leave the flexibility open for those feature matrix extensions. And additionally, because our goals were different, the guidelines and process for the validation were also a little different. We once again relied heavily on EPA and FDA guidelines to ensure that we would meet our method purpose and still maintain that flexibility. So for example, a significant part of our validation was the use of blind spikes to demonstrate the ability that an analyst could correctly identify all dyes that were present in an unknown sample. And just for fun, these chromatograms on the right, we obtained during feasibility. So we have a profile from a grape candy, which contained blue 2 and red 40. And we also have a ranch flavored corn chip extract, which had yellow 5, red 40, and blue 1. And since the development and validation of this method, we have had many unique and interesting applications. So I wanted to share a couple more here. So our first, we also have used this method on a discolored soup concentrate investigation. So we had consumers reporting a soup concentrate with an off-putting beige color, and the expected color was bright yellow. So this is the control sample on the left, and you can see that the sample contained low levels of yellow 5 and also yellow 6 and red 40. And then the suspect, as you can see, it really is quite a beige color, not very pleasant, and this soup sample was then lacking a significant amount of those dyes that should have been present. Another case study that we used this method for was a fruity drink that had an off odor and off color. A consumer noticed a discoloration in their orange flavored drink, and it had an unpleasant aroma, so they were concerned about potential contamination. So this is the profile of our control sample. You can see the nice orange color as expected, and we saw yellow 6. And then in our suspect, which had almost a greenish color, we saw a significant amount of blue 1 dye as well. So in this case, the dye screen was used as a part of this investigation. We also actually did Carl Fisher, we did metals analysis, and we did GC-MS screening on this sample. And we ended up finding isopropyl alcohol and an assorted phenol compounds as well by GC. So putting all of this information together, we were able to infer that this sample had been contaminated with what was likely to be some kind of blue cleaning product that was IPA based. So just to summarize what we've gone over today, validated analytical methods are essential to ensure accurate testing of products that have a significant impact on human health and safety, such as foods and pharmaceuticals. In order to successfully develop a method with the goal of validation, it is important to first consider the goals of the analysis and also the properties of the analyte in order to choose a platform that is likely to give good results. If you consider all of these factors before beginning and use that in tandem with literature and institutional knowledge and other published methods, this process, which can seem overwhelming, will be much smoother. And here are some references and helpful resources that you can use later on when you receive a copy of these slides. At this time, I'll hand it back over, and we'd like to thank you all for attending this webinar. We'd be happy to answer questions that you may have.
Perfect. Thank you so much, Lily. So we do have a few questions in the chat. So the first one we have is, what makes an analyte more or less challenging when developing a method?
Yeah, I can take that one. So what makes something more or less challenging, it depends on a number of different factors that can impact method development difficulty. For example, if we're looking at the analyte specifically, having the presence of a lot of analytes with very similar structures in your product can make it more difficult, as you need to find a way to separate those effectively. However, a lot of other factors other than the analyte itself can play a role in method development difficulty. A big one is sample matrix. So for instance, it's a lot easier to develop a method for something like water than it is to, say, for something like cheese or a cookie that has a lot of fat or protein present. So in those cases, you might have to look for more complex method cleanup or separation in order to achieve your goals. Similarly with, for example, needing lower limits of detection, in those cases, you typically have a lot more matrix interference issues and need to be a lot more specific and a lot more careful with your matrix cleanup.
Great, thank you so much. Another question from the chat: if you have access to a mass spec, why wouldn't you use that for most methods, given the specificity and sensitivity?
Sure, so I can take that one. So as we talked about, there are benefits and drawbacks to every system, and it always comes back to thinking about what your analytical goal is. So one of the biggest drawbacks for a mass spec, both GC and LC, is that it is more expensive to run. It's more time intensive, it's more complex, and additionally, there are times when amazing selectivity and sensitivity aren't actually going to give you what you need, and it could even be detrimental to the development, such as for an assay method. So mass specs are great for the right application, but as a whole, our default is typically to use something cheaper and simpler unless we have a reason to believe that we need that extra layer that the mass spec can provide.
Great, thank you, thank you. And then we have another question in the chat. Why would you need to change a method if one already exists for raw material?
Sure, I can take that. So actually kind of relates back to the first question where I talked about sample matrix. But when you're looking at a raw material, you're typically looking at something that is fairly pure and fairly concentrated. So when you have a method for that versus a method in a final product, you're typically needing to consider greater factors such as matrix interference, lower concentrations, and so often you need to update or in some cases completely develop a new method in order to separate out your analyte from those matrix interferences and look for lower concentrations. And so there's a lot more to consider when working with a complex finished product.
Perfect, thank you so much. All right, well, thank you so much, Jamie and Lily, for today's presentation, and just wanted to extend a big thank you to everyone who's attended. Please feel free to reach out with any questions, and we would be happy to connect further to discuss this important topic. This now concludes today's presentation. I hope everyone has a wonderful day.